Around the world, scientists are closely watching the emergence of several variants of the coronavirus. These variants are causing concern because they contain mutations in the spike protein, making them potentially more infectious and more deadly. Dr. Michael Peters is a professor in the Department of Chemical and Life Science Engineering at Virginia Commonwealth University. Dr. Peters says knowing how the spike protein is changing is key to understanding how the virus works and can offer clues about the next possible threats. Dr. Peters, thanks so much for your time today.
Thank you.
You conduct experimental and theoretical research in the field of protein engineering. One of the major mutations seen in the UK variant is to that of the spike protein. So, why is spike protein such an important target of research, and what makes this mutation significant?
Yeah, so you probably have seen all the cartoons of the SARS-CoV-2 virus, and it shows these little red fuzzy balls all over the surface. And that's not too far from reality. Those fuzzy balls are the spike protein. So it densely decorates the entire surface. So if you're looking at it, and if your immune system is looking at it, the first thing it sees is the spike protein. It doesn't see much of anything else. So that means then that as far as when we're talking about vaccines, if we can present our immune system with just those fuzzy balls and not the whole virus, then our immune system will effectively take pictures of those fuzzy balls. And then hopefully when the real virus comes into your body, your immune system will line those pictures up of the fuzzy ball with the actual virus and say, hey, we got a problem here. Let's take this out. So the spike protein becomes critical then in the development of vaccines. And then it's also being used in targeting therapies because the virus uses a spike protein to invade cells. Its initial attachment to our epithelial cells is with these fuzzy little balls. And so that's why the spike protein is so critically important.
And you explain it in such layman terms. The research is very complex, but we're learning that the virus stabilizes itself in an up or down state. So, what has your research yielded in this area, and how can this be key to the development of those therapies you were talking about?
Yeah, so that's a very good question because a lot of this is still being studied mechanistically and scientifically. So basically, the spike protein is actually three proteins together, and so these proteins are sandwiched. Three of them are sandwiched together, and actually in a cylindrical fashion. And so, so think of raising your arm up and your hand up. Well, one of these proteins could be in the up state with its arm up and its hand up, or it could be in the down state where its hand is down. And so it's believed that if the hand is up and the arm is up, that it can latch on to our epithelial cells a lot easier than if it's down. So consequently, a lot of effort is being made to try to understand this up versus down state and what that might imply, not just in terms of vaccines, but also therapeutics and also variants that may involve, so mutations of the spike protein are associated with variants. And so what do those mutations do to this up versus down state? So that's what we're looking at, and it has importance across vaccines, therapeutics, variants of concern, transmission rates, and almost everything to try to understand how this virus works.
Well, it is suggested that we're in a race between science, vaccines, and variants. Is it important to study all of the variants? How do researchers determine what are the most important variants to study to stay ahead of the virus?
Yeah, so it's really the CDC that tracks data to establish what they will call either a variant of interest which has a potential um, um, detrimental impact on vaccines or therapeutics or higher transmission rates, or a variant of concern where the data shows it's having an impact. So for example, the UK variant, which the UK doesn't like us calling it the UK variant, it's B.1.1.7. And then there's the South African variant, which is the B.1.135, I think. But those variants then, are associated then with mutations of the spike protein, and they do cause, as the data shows, increased transmission rates. And then there's a third classification, which fortunately we're not in right now, which is called a variant of high concern. And again, that's data driven, where you have a more severe impact on the population, increase in mortality, higher rates of transmission, and so forth. So all this is established by the CDC. And what's neat about the CDC is they're data driven. They go by data, and they monitor data all the time. They're looking at sequences, they're looking at the prevalence of sequences, and they do some pretty sophisticated statistical analysis all the time to try to come up with better guidelines. But I think sometimes the public doesn't understand that they're data driven, and they're very reluctant to come out and issue a new guideline or do anything unless they've got the data to back it up. So the good thing from my point of view, when I study the possible changes from a biophysics point of view, is that I accept what the CDC provides statistically. So if the CDC says, look, this new variant, I think it's the 484 that they're looking at now, okay, that's a variant of concern. Well, I'm going to start looking at it because I already know that the CDC has done the statistical analysis and knows that that's potentially important. So that's really kind of cool, really.
Well, and when it comes to developing therapies and vaccines, I guess we all need to understand how the immune system works. So if you've had one variant, would you have antibodies against another? How does our immune system respond to this virus?
Yeah, so that really is a complicated question. I always use the metaphor of this sort of perpetrator breaking into your home, right? And so the virus is the perpetrator, your home is your epithelial cells. And so, we know the vaccine will take a picture, right, of this spike protein, and then it'll kind of line it up if your perpetrator comes into your house. Well, what happens then is that, of course, the perpetrator, every time it invades somebody's home, it has an opportunity to disguise itself for the next break-in, and that's the mutation. So the virus cannot disguise itself unless it invades your home. So it invades your home, it goes through its whole process, and then it can put on a new disguise to go to the next home. So each time it transmits, it has a possibility of disguising itself more and more. So one time it could put on a mustache, and then next time it invades, it could put on a blonde wig or whatever, sunglasses. And so as it continues to mutate, then your immune system is going to have a harder and harder time to line those pictures up and say, is that really the one? And so therein lies the problem. That's called mutational escape. That's actually a formal virology terminology, mutational escape, where now your antibodies no longer are effective, which are basically your pictures, your memory cells, and antibodies are your pictures. And that pictures don't line up very well anymore. So that's what we're worried about now, because we have a vaccine and we see the light at the end of the tunnel. But now more than ever, we've got to prevent the virus from infecting, because each time it infects, it's going to put on a new disguise. And we already know it's wearing some disguises right now. but fortunately the vaccines are so robust that they can pick it up. So the virus hasn't outsmarted the vaccines yet. So the virus has put on some sunglasses and a mustache, but right now these vaccines are so effective that they're saying, that's still the spike, that's still the SARS-CoV-2. We're not fooled by that, you know. But if you keep letting it add more disguises, you know, puts on a big coat, and you know, before long our immune system is like, I don't know what that is. And so that's why it's so important right now that we double our efforts to mitigate. So mask, distance, ventilation, and active filtration. Those are the things that we really need to double down on now, because we've got the light at the end of the tunnel. And if we let the virus keep changing, then we're shooting ourselves in the foot with this. And so that's what a lot of people are worried about. And that's why you see that there's so much controversy and talk in the news about states relaxing mask mandates and so forth. This is not the time to do that. It's shooting yourself in the foot. It's really, really a dumb thing to be doing when we have a vaccine. That's the ironic, you know, part of it. We have an effective vaccine. Why do you want to take a chance like that? So thank you for that question.
And thank you for the analogy. It's a great one. So what you're saying is that what the data is bearing out is that the efficacy of the vaccines rather that are currently available are effective against the variants to date?
Absolutely. Absolutely. Yeah. And so, you know, that's why people need to go out and get vaccinated. You know, it's going to keep you out of the hospital. It's going to keep you, you know, alive. So absolutely, they're still effective. And so we have a good opportunity here if we don't blow it.
Well, we really appreciate you sharing your expertise and in the simplest of terms. And also, we thank you for the important research that you are doing in the battle against this virus. We really appreciate you sharing that with NewsPoint 360 today.
Sure. Thank you.